Hello children, welcome to the class. Today we are going to discuss about plant hormones, which forms a part of the chapter Chemical Coordination in Plants. Now as we know that hormones are chemical messengers secreted in the body of animals as well as the plants. They may be secreted in one part of the body, they travel through the body, reach another part, and always they show their impact on their target cells or target organs. Now the first hormone to be discovered was secretin, which is a gastrointestinal peptide hormone, and the credit for its discovery goes to two scientists, William Bayliss and Ernest Starling, in the year 1902. The term hormone was also coined by these two scientists. Now, like the animal hormones, the plant hormones are also secreted naturally in the body of the plants. All the plant hormones that we will be discussing today, they are, as we can see, auxins, gibberellins, cytokinins, ethylene and abscisic acid. These plant hormones were also given the term as phytohormones to demarcate them from the animal hormones. The plant hormones are also known as growth regulators. The first three hormones mentioned here, they are also called the growth promoters, and the next two, the ethylene and abscisic acid, they are also known as the growth inhibitors. So as the term implies, promoters will of course promote the growth of the plant body and inhibitors will try to stop the growth. Now, plant hormones are also available in their synthetic forms, and these synthetic forms of the hormones are used worldwide by different agriculturists, horticulturists, for various purposes like increasing the yield, the crop, the fruits number, etc. Now the discovery of the first plant hormone, that is auxin, the credit goes to few scientists. The first name here, that is Charles Darwin, and his son Francis Darwin in the year 1880. Later another scientist by the name of Peter Boysen Jensen in the year 1910 and 1913 also performed experiments on the plants. And finally it was Fritz Warmolt Wendt in the year 1928 who did his experiments on the plants. The first three scientists, they performed their experiments on the canary grass coleoptile. And Wendt performed his experiment on the oats coleoptile, which is also, oats are also called avena, so it is also known as the avena coleoptile. So what these scientists did, we will discuss about the experiments, but first we should understand what is a coleoptile. So let us recapitulate few things from the chapter seeds. We have seen that in the monocot seeds, inside the seed, in the embryo, the shoot apex is always covered by a protective sheath known as the coleoptile. So when this seed starts to germinate and it forms the seedling, it is the coleoptile which comes out first, the cylindrical structure, in the form of a cylindrical structure. It comes out first and pushes the soils and comes above the soil. And it starts growing and becomes taller and taller. This coleoptile protects the delicate shoot apex while the seedling is coming out of the soil. Now children, here I am drawing the coleoptile. Like I draw here three coleoptiles I am drawing over here. I name them as A, B and C. Okay. So the first experiment conducted by these scientists, in the first case this coleoptile is growing taller and taller. So what they did, they first cut the apical part of this coleoptile and just they tried to observe, like for example I draw it over here, and they observed whether it will be growing or not. So they found that after the removal of this top part, this coleoptile here, it stopped to grow. So this confirmed that some kind of chemical is present in this apex of the coleoptile over here, which I am drawing by these circles, which moves in the downward direction and helps in the growth of this coleoptile. And in their second experiment, they exposed this coleoptile to a source of light. This was placed in a dark room, exposed it to a source of light. Now their finding was that after some time, this coleoptile which was growing, it started to bend towards the sun light or light source like this. So they concluded that this chemical substance which is present in this coleoptile, it moved towards the side of the coleoptile which was towards the dark from the part which was exposed to sunlight. And they also concluded that the part of the coleoptile which is exposed to light, it reduces the synthesis of this chemical substance auxin. This term auxin was coined by Wendt later on, and as the auxin becomes concentrated toward the darker side, this side grows more when compared to the side which is exposed to sunlight. So now here you will tell that teacher, the plants grow toward the source of light. This is called phototropism. Of course you are right. The stimuli light helps in guiding this shoot to move towards the light. But apart from that, the auxins also play a very important role in the bending of this shoot apex towards the source of light. So that is how the discovery of the auxin took place, the first plant hormone. Now let's just go through the functions of this hormone auxin. The first function that we saw here, it helps in cell elongation. Auxins we will normally find in the shoot apex and the root apex, the meristematic part of the plants, and helps in the apical elongation. And the presence of auxins, it will just it will increase the flexibility of the cell walls of the cells, the meristematic cells present in the apical region, and it helps in the enlargement of cell. That is how it helps in the apical growth. Second important function of auxins is apical dominance. Now when the plant is growing and these auxins are present over here, it will only allow the apical growth. It will go on increasing, it will become taller and taller. But these auxins inhibit the growth of lateral buds. It will not allow the lateral buds to come out. That is called apical dominance. Third important function is it delays the leaf senescence, does not allow the leaves to die, keeps them alive and young for a longer time. Next one, induces parthenocarpy. Presence of auxins in the fruits, in the plant, it without fertilization, it helps in the formation of fruits. So those fruits will be formed without any seeds. Like nowadays we have the seedless grapes and certain other fruits also. It induces rooting in the stem cuttings. Like children, we like to grow the stem cuttings of rose, bougainvillea, china rose, etc. So if in the tissue, the culture medium where we insert the stem, if we add auxins, it will induce the rooting. The rooting will become faster. Controls the abscission layer. Like abscission layer is a layer which is formed just at the end of the petiole in the leaves or in the pedicel of the flowers and fruits. So it inhibits the formation. So when abscission layer is not formed, it reduces the fruit drop, flower drop, leaf drop, etc. So flowers, fruits remain attached to the plants for a longer time. It reactivates the vascular cambium, the cambium present in the vascular bundles, and it also helps in tissue culture and callus formation. This tissue culture and callus formation is a form of vegetative propagation. So when those cells are, somatic cells are extracted from the plant body, kept in a culture media, if we add auxins over there, it helps in the formation and enlargement of more cells. So these are the functions of the auxins. Now we come to the second hormone, gibberellins. This plant hormone was discovered by Kurosawa and it was extracted by him from a fungi known as Gibberella fujikurae, which was causing foolish seedling disease in the rice plants in Japan in the year 1926. Later on, many varieties of gibberellins were identified, over 100 different varieties were identified from different plants, and approximately 70 different types of gibberellic acids were extracted from different plants. And they were named as gibberellic acid 1, gibberellic acid 2, gibberellic acid 3 and so on and so forth. Out of all these, gibberellic acid GA3, this is the most studied gibberellin of its types. Now this gibberellin, it also helps in the growth of the plant, and it generally works in the internode area, children, that is to be remembered. It is secreted in the internode area and helps in the elongation of the stem by the elongation of the cell. So that is how it affects the growth of the plant. It also helps in the leaf expansion. It helps in a very important process called bolting of dwarf plants. We have all seen the cabbage plant where between the cabbage leaves we have seen that very less internode space is there. But cabbage, just before flowering, it all of a sudden becomes very tall and the internode becomes very taller, or large internodes develop because of the secretion of gibberellins at that particular time. So gibberellins can also help in the reversal of the genetically dwarf plants. If it is applied to genetically dwarf plants, it will increase the length of the internodes and increase the height of the plant. Next is it breaks the seed dormancy. Seeds which are not germinating, seeds which have a very thick seed coat or don't have the proper conditions available. So by application of gibberellins, it can initiation of the seed germination takes place. Gibberellins also help in the initiation of germination in the dormant buds and tubers. It helps to promote fruit growth. Now we have seen the long grapes, thinner ones and longer ones. So application of gibberellins helps in increasing the length of the grape. Gibberellins can also help in increasing the length of apples and giving them a proper shape. And because of this, it can induce parthenocarpy also, that is, it can induce fruit formation without fertilization, that is, without seed formation. So we love to eat the longer grapes without any seeds. These grapes actually they are known as the Thompson grapes. Because of gibberellins we get these kind of grapes. Now speeds up malting. Malting is an important process in the brewing industry, and here we should understand what is a malt. Malt is a cereal grain which undergoes soaking treatment and then it is allowed to germinate. And as it is germinating, it is exposed to hot air to stop the process of germination. Now by this process of malting, a large number of enzymes are produced by these cereal grains. The most important cereal to be used for malting is barley, but other than barley, wheat, rice, oats, all these also can be used. The enzymes which are produced are amylases, proteases, which can break the complex food like the starch, the proteins in the grains and convert them into the fermenting sugars. So that is how. So malting process takes 7 days or more than that, but gibberellins can help in reducing that malting period to 2 to 3 days. And another important function is vernalization. Now vernalization is a low temperature treatment given to certain plants like chrysanthemum and henbane to induce flowering in them. But without the low temperature treatment also, gibberellins can induce the process of flowering in them. Next is the cytokinins, another important hormone which many scientists detected its presence in different parts of the plant, but finally it was Skoog and Miller who said that these chemicals they promote growth by the process of cell division. So we know that already all of us know that cyto means cell and kinesis. Kinesis means division. So it promotes growth by cell division. Now, okay, that is very important function of the cytokinins. Then it also helps in the expansion of cotyledons in the seeds, break the seed dormancy, initiates germination in the seeds, delays the leaf senescence, death of the seed, death of the leaf is delayed and the leaves can remain attached to the plants for a very longer time and they can remain young for a longer time. Then it promotes the chlorophyll synthesis in the chloroplasts of the plants, green cells. Inhibits apical dominance. Now you see, auxins promote apical dominance. They don't allow the lateral buds to grow. But cytokinins will stop the apical dominance and it will allow the lateral buds to grow. This is just opposite to the auxins. And the cytokinins which are secreted in the roots of the plants and travel to various parts of the plants to help in the growth over there. They also help in the process of morphogenesis. Now morphogenesis, we saw in the case of auxins that auxins can initiate help in the cell growth, cell elongation in a culture medium, where the parenchymatous cells are obtained from the plants and kept over there for tissue culture. So there cytokinins are also added in this tissue culture. So both auxins and cytokinins together, they help in the division of the cell and the growth of the cell, and hence they help in the formation of callus, from which many explants can be extracted. The last two plant hormones, they are also known as the growth inhibitors. And out of this, ethylene is the gaseous hormone, the only gaseous hormone. Ethylene was recognized as a plant growth regulator by two scientists by the name of Galston and Davies in the year 1970, and it was recognized to be ethylene by a scientist by the name of R. Gain. Now children, in the previous time also, people used to observe that whenever raw fruits were exposed to smoke or they were exposed to fumes coming out of burning substances, the process of ripening used to enhance in the fruits. Today also we sometimes insert the raw mangoes in hay or keep the raw mangoes under rice in the rice drums just to enhance the process of ripening. So we have understood that ethylene plays a very important role in the ripening of the fruits. Another important aspect of this ethylene is that during the process of germination in the dicot seeds, we saw that in the monocot seeds, the coleoptile is present, which protects the shoot apex while germination, while its emergence from the soil, and later also it plays important role in the dicot seeds. When the dicot seed has germinated, the plumule and the radicle has come out, and you can see here that the hypocotyle has emerged, and when the hypocotyle of this seedling reaches the soil, and it has to break up the soil and emerge out of the soil, and when this puts pressure against the soil, at that time ethylene is secreted in this shoot apex of that hypocotyle part, and this helps in the formation of a hook-like structure which is known as the apical hook. Now after it breaks the soil, it comes out of the soil and in this form. At this point, if this apical hook is supplied with ethylene gas exogenously from outside source, it will keep on growing like this horizontally. But naturally this does not happen because we are not supplying ethylene from any outside source. So once it has come out like this, now it will become erect like this, and it will start growing in the upward direction and also towards the light. Another important function of ethylene is that it is a powerful inducer of aging and senescence, that is death of the cells. It inhibits the elongation of stems and roots because it is a growth inhibitor. But children, sometimes this ethylene can also act as a growth promoter. So as you can see here that it initiates germination of peanut seeds, initiates sprouting of potato tubers, and it can also induce flowering in the mango and pineapple plants. But whereas it retards this process of flowering in most of the plants, prohibits them. Now we come to abscisic acid, which is abbreviated as ABA, and it is a very important stress hormone for the plants. Abscisic acid is a very strong growth inhibitor, and it reduces the metabolism taking place in the cells of the plants, mostly found in the fruits, vegetables of the plants. Now it inhibits the growth. The credit for the finding out about abscisic acid goes to Frederick T. Adicott and Larry A. Davis in the year 1963. Now coming to the functions once again, it induces seed dormancy and it inhibits the seed germination, just opposite to gibberellins. Then it accelerates aging and senescence. The cells will become aged fast, they will die, and that can lead to the fruit drop, leaf drop, flower drop very easily. It also enhances the process of formation of the abscisic layer at the base of the pedicel, at the base of the petiole. Acts as a stress hormone just like I told you just now. So it protects the plants from any different kind of stressful conditions like drought, like salinity, and it is of very much advantage to the plants which are growing in the desert regions. So whenever there is scarcity of water, so this hormone, it makes the stomata present in the epidermis of the leaves of those plants to close, so that loss of water is prohibited. Again, it suppresses the cambium activity in the vascular cambium of the plants. So these are the different functions of the various plant hormones, children, and hope you have understood the concept well. And thank you for watching the video.